So we're going to do chapter 9. We're going to talk about cell division, all the C words like chromosome, chromatin, centromere, sister chromatid. We'll talk about gametes versus somatic cells, binary fission, the phases of the eukaryotic cell cycle, and cytokinesis and how it's different in plants and animals. We will also talk about controlling the cell cycle, checkpoints, and tumors, and like a tiny bit on cancer as well. So all cells come from pre-existing cells, and even multicellular organisms are produced by a series of repeated rounds of cell growth and then division. And really, cell division is a highly regulated series of events. And eukaryotic organisms, so cells with a nucleus, cell division occurs by mitosis or meiosis. And in prokaryotic organisms, it occurs by binary fission. So our first picture up there is cell division in animal cells, then we've got plant cells, and then we've got bacteria. So binary fission in prokaryotic organisms, E. coli is an example of a prokaryotic organism, so a bacterium. In E. coli, the chromosome replicates beginning at the origin of replication, so we make a copy of the chromosome there, and then the two daughter cells are going to actively move apart while the cell elongates and the plasma membrane pinches inward, and it divides the one cell into two new identical cells. So that's how prokaryotic organisms divide. So as far as organization of genetic material, a genome is all the genes in a cell, and those genes are on DNA, and that DNA is packaged into chromosomes. Prokaryotic cells have a single, often circular chromosome, and eukaryotic cells have a number of linear chromosomes that are packaged into chromatin, which is found inside of the nucleus. Not all eukaryotic cells have the same number. For example, dogs have 78 total, so 76 autosomal chromosomes and 2 sex chromosomes. Fruit flies have 8 total, and adders, tongue ferns have 1,260 total chromosomes, which is a lot, right? So then we can talk about cells being either haploid or diploid. So haploid cells have only one set of chromosomes. So animal gametes like sperm and egg or plant gametes are haploid, and also the kind of body cells that make up a fungus would be haploid as well. Then diploid cells have 2 sets of chromosomes. For example, animal body cells or the sporophyte generation in plants would be diploid. So when eukaryotic cells divide, the chromosomes become compact enough to be seen with a light microscope, and a karyotype basically sorts these out and reveals the number and size and the form of the chromosomes and an actively dividing cell there. So there's an image of a karyotype on this slide. So was the karyotype on the previous slide taken from a diploid or a haploid cell? So go back and look at that image. And if you look, you can see that there's 2 sets of chromosomes. There's 2 chromosome 1s, and really there's 2 chromosome 1s and their copies. So that's going to be from a diploid cell. And then in humans, what type of cell only needs half the genetic information of a normal cell? So that would be sperm or egg that only needs half the genetic information. So sperm and egg in humans are haploid. And generally speaking, sperm and egg are haploid. So that brings us to homologs or homologous chromosomes. So diploid species have homologs, which are pairs of chromosomes. Like you saw in that karyotype, there were 2 chromosome 1s and 2 chromosome 2s and 2 chromosome 3s and so on and so forth there. So the first image we have on the slide, you've got 2 chromosomes that carry the same kinds of information, but they might have a slightly different version of that information. So they have the allele for purple flowers and the allele for white flowers on the other chromosome. But they both have the gene for flower color. So they have the same kinds of information, it just might not be identical. And we'll talk about that more in the next unit. And then as far as autosomes go, each homolog is nearly identical in size and genetic composition. Again, they both have the gene for eye color, but one may have the allele for brown and the other has the allele for blue. And really, eye color is much more complicated than that, and we'll talk about that a little bit later coming up as well. So that brings us to sex chromosomes. Sex chromosomes can be very different in size, and they can be part of a sex determination system. So in humans, we've got X and Y chromosomes. So XX is female and XY is male. But that's not the only sex determination system possible. Fruit flies are also XX and XY, but chickens are different, and so are pigeons. So that brings us to the eukaryotic cell cycle. I feel like most of you have probably gone over this at some point in time before. But generally speaking, eukaryotic cell cycle includes interphase, which is made of G1, S, and G2, and then M phase, which is mitosis and cytokinesis. And then mitosis is further broken down into prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis. Some cells have G0, which substitutes for G1 in cells that are going to no longer divide. I also think it's important that you look at the image to the right. We're going to spend more time talking about mitosis because there's more things that you can see under the microscope during mitosis. We even have a lab where we look at slides and identify the different stages of mitosis. However, it's important to realize that the majority of the cell cycle is spent during interphase. So G1 is the first phase of interphase in the eukaryotic cell cycle. In this phase, the cell is growing, and in a way it's like growing to recover from the previous cell division. In this phase, signaling molecules can cause the cell to accumulate molecular changes that promote progression through the cell cycle. If the cell passes this restriction point or G1 checkpoint, the cell becomes committed to enter S phase and replicate DNA. If the cell does not receive that go-ahead signal, it will exit the cell cycle and switch into that non-dividing state called G0. So that brings us to S phase. This phase, the cell prepares for cell division and the DNA replicates. So the S you could think of as synthetic or synthesis phase for DNA synthesis. The duplicated chromosomes are going to stay joined to each other, and now we call them sister chromatids. And where they are most closely attached is called the centromere. So that brings us to G2 of interphase. This is the next growth phase, and really in G1, S, and G2, the cell is going to continue to grow throughout all of those. But in G2, the cell produces proteins that are needed during mitosis and cytokinesis. And a human cell in G1 would have 46 chromosomes, and the same cell in G2 would have 46 pairs of sister chromatids, or 92 chromatids total, because in the previous phase, S phase, they replicated the DNA. So that brings us to M phase, which technically includes mitosis and cytokinesis. So mitosis is when we divide the nucleus into two identical nuclei with the separation of sister chromatids. Mitosis could be used for asexual reproduction and growth and repair. And the phases of mitosis are prophase, prometaphase, metaphase, anaphase, and telophase. Then cytokinesis is when the cytoplasm actually divides, and it usually follows mitosis. There's another kind of cell division which we'll talk about in the next unit in eukaryotic cells, and it's called meiosis or meiosis. And this is going to produce non-identical daughter cells that have only one set of chromosomes and have half as many chromosomes as the parent cell, which will be important for sexually reproducing organisms. We'll talk more about that in the next unit though. So on to the first phase of mitosis, which is prophase. In prophase, the chromosomes have already replicated and are joined as sister chromatids. Remember that they replicated in S phase of interphase. The nuclear membrane dissociates into small vesicles, and the chromatids are going to condense into highly compacted structures that are readily visible under the microscope. Prometaphase may be one that you didn't hear about in grade school if you remembered like prophase, metaphase, anaphase, telophase, or PMAT. So I guess now it's PPMAT. But prometaphase is kind of this in between of prophase and metaphase. So the nuclear envelope is completely fragmented at this point. The mitotic spindle is fully formed during this phase. The centrosomes are going to move apart towards the end of the cell or towards the poles. The spindle fibers are going to interact with the sister chromatids, and then there's two kinetochores on each pair of sister chromatids, and they're going to attach to the kinetochore microtubules from the opposite poles. From there, we've got metaphase, which I think is the most recognizable phase of mitosis. In metaphase, the pairs of sister chromatids are going to be aligned along the cell, kind of in the middle. We call that middle of the cell the metaphase plate, or sometimes we call it the equator. And they're organized in a single row, and when the cell alignment is complete, the cell is said to be in metaphase. So I think metaphase, middle. In anaphase, the individual chromatid is going to be linked to only one pole by the kinetochore microtubules, so the sister chromatids are separating, and the microtubules are pulling the chromosomes towards the poles. So as far as remembering anaphase goes, I think like the sister chromatids are making these little A's, or the chromosomes are making these little A's, or imagine as they're being pulled apart, they go, ah! However you remember anaphase, that's up to you, but there's a couple ways that might help you. So that brings us to telophase or telophase. In this phase, the the chromosomes have reached their respective poles and they have decondensed, so they've relaxed, and the nuclear membranes reform and produce two separate nuclei. So basically we're putting the cells back together in telophase. So then remember, mitosis was the division of the nucleus. Usually that division of the nucleus is followed by division of the cell itself. So cytokinesis is that division of the cytoplasm. So here, the two nuclei are going to be separated into separate daughter cells. This process is different in plants and animals. Animals form a cleavage furrow, which constricts kind of like a drawstring, and that separates the cells and pinches them apart. Plants have a cell wall, so you can't really pinch them apart. So instead, they have to form a cell plate to build a new cell wall between the two daughter cells. So here's kind of an example of mitosis in the onion root cell, and I think it's important that you're able to identify the different phases of mitosis. So this first image here that I've drawn a box around would be a cell in metaphase. And then what do you think this phase is? That one's maybe tricky. That one is actually interphase, so it's not in mitosis. It's just hanging out doing regular cell things, so that's interphase. This next one I would say is telophase or telophase, however you want to say it, no big deal. From there, this would be anaphase, and then that one would be prophase. So make sure you could look at a picture. We'll look at these in lab, be able to identify the phase of the cell cycle by a description or by an image as well. So as far as regulating the cell cycle goes, it really depends what kind of cell you are, how quickly or how frequently you will divide. So, you know, if we think about skin cells, your skins are under kind of attack from the environment all the time, so we have to regenerate those readily, whereas the cells in your brain don't divide as readily. So the difference really results from regulation at the molecular level. There's a couple of different ways we can control what's happening in the cell. The cell can control itself based on internal signals or external signals. So some internal signals include checkpoints like kinases and cyclins, and then external signals include growth factors, density-dependent inhibition, and anchorage dependence. So as far as internal control of the cell cycle goes, there's 3 checkpoints in eukaryotic cells: G1 checkpoint or the restriction checkpoint, G2 checkpoint, and the metaphase checkpoint. Checkpoint proteins basically act as sensors to determine if the cell is in good enough condition to divide. And the cell cycle is delayed until the problem is fixed or prevented altogether. And loss of checkpoints can lead to mutations, which are changes in the DNA, and that can ultimately lead to cancer. So this diagram does a good job showing you the various checkpoints. That first checkpoint is the G1 checkpoint, or the restriction point, and it checks to see if the DNA is damaged. So G1 cyclin is made in response to sufficient nutrients and growth factors. And so the G1 cyclin binds to the cyclin-dependent kinase. And then we go on to S phase. In S phase, we replicate the DNA. The G2 checkpoint determines if all the DNA is replicated, and it monitors the levels of proteins needed for M phase. So when the mitotic cyclin binds to the cyclin-dependent kinase, we've met the G2 checkpoint, and we can go on, and mitotic cyclin is degraded as the cell progresses through mitosis. Then we get to the metaphase checkpoint. This makes sure that all the chromosomes are attached to the spindle apparatus. So those are the eukaryotic cell cycle checkpoints. So there are also external controls of the cell cycle, like growth factors. Growth factors are proteins that are released by certain cells that stimulate other cells to divide. For example, platelet-derived growth factor stimulates the division of human fibroblast cells in tissue culture. So in this image, we've got a sample of human connective tissue that's cut into small pieces in a petri dish. There's enzymes that digest the extracellular matrix, resulting in a suspension of free fibroblasts, and the cells are then transferred into culture vesicles. And without platelet-derived growth factor, the cells do not divide readily, but with platelet-derived growth factor, they do readily divide. We also have density-dependent inhibition and anchorage dependence. Density-dependent inhibition is when crowded cells stop dividing, and anchorage dependence is when cells have to be attached in order to divide. So anchorage dependence, again, is when cells require a surface for division, and then density-dependent inhibition is when the cells form a single layer, and if we take out a chunk of those cells, they would fill the gap and then they would stop dividing again. So this is important in the formation of multicellular organisms. So cancer cells don't respond to signals that normally would be regulating the cell cycle. They exhibit neither density-dependent inhibition nor anchorage dependence. They also do not need growth factors to grow and divide. They may make their own growth factor. They may convey a growth factor signal without the presence of a growth factor, and they have some sort of abnormal cell cycle control system. And the process that converts a normal cell to a cancerous cell is called transformation. So as far as spread of cancer cells go, cancer cells that are not caught by your immune system form tumors. A benign tumor is a tumor that remains at the original site, and they really maintain most of the original tissue characteristics. So the cells are going to stick to each other. Malignant tumors, on the other hand, invade surrounding tissues and undergo metastasis and may form additional tumors. So chapter 9 is relatively short. We talked about cell division just kind of in general. We talked about chromosomes, chromatin, sister chromatids, centromeres. We talked about gametes versus somatic cells. So gametes are haploid, somatic cells are your normal body cells, and they're diploid in organisms such as ourselves. We talked about binary fission, which is the cell division in prokaryotic organisms. We talked about the eukaryotic cell cycle. So we'd have prophase, which is G1, S, and G2, and then M phase, which is mitosis and cytokinesis. And with mitosis, we have prophase, prometaphase, metaphase, anaphase, and telophase. We talked about cytokinesis in plants and animals. Remember, there's a difference. Plants can't pinch off the way the animals do, so plants have to build a cell plate in order to build their cell wall. And we talked about cell cycle control with internal and external factors regulating the cell cycle. We talked about checkpoints, and then a little teeny tiny bit on tumors and cancer. So that is the end of chapter 9.